Welcome everyone to this webinar on structural biology data processing workflow pipeline automation using AI/ML. My name is Devan Atnur. I am VP of Scientific Strategy at Clovertex. With me, we have two professors, Professor Daisuke Kihara from Purdue University and Professor Toshio Moriya from KEK from Japan. What we are going to cover today is really to talk about the present state of cryo-EM data analysis. What are the opportunities of automation in the cryo-EM workflow for data analysis? We will touch upon single particle analysis, compound screening, some of the atomic model building, how deep learning helps in automation, and how cloud accelerates this automation. So without further ado, we'll start with introductions. Toshio-san.
Thank you. Thank you for introduction. So my name is Toshio Moriya from High Energy Accelerator Research Organization in Japan. The abbreviation is KK, but this abbreviation is from the Japanese name, Kokasoku Energy Kenkyujo. So it's a kind of weird abbreviation, but the KEK is very catchy. I really like it. So please just remember that it's a KEK and everybody use it. So the KEK is one of the major synchrotron facility in Japan. It was the oldest, and now that it's getting a little bit old, but it's very active. And we have the two institutes inside of KK. One is Institute of Material Structure Science and Accelerator Laboratory. So I belong to the Structural Biology Research Center, which belongs to the Institute of Material Structure Science, and we are doing the biological research. And so we have two major facilities. One is Photon Factory. This is the original facility that we are running and supporting all the Japanese researchers who are doing the X-ray crystallography and the SAC, small angular the X-ray crystallography. The angular scattering. And then we recently introduced also cryo-EM for the sake of the structural biology research, because this became really practical. Cryo-EM became practical for the solving the structure of the protein. So that is what we do. Thank you, Devin.
Thank you, Toshio-san. Professor Kihara.
Okay. Hi, I'm Daisuke Kihara. I'm professor in biological sciences and computer science at Purdue University. What my lab does is the tool development for protein structural modeling for cryo-EM. This is my main project right now. I have a background of protein 3D structure prediction, protein-protein docking prediction, computational drug screening. We also do genome annotation from amino acid sequence, and we develop tools for this. So today I will introduce a couple of tools we have recently developed for protein and DNA, RNA structure modeling for cryo-EM maps. Thank you.
Thank you, Professor Kihara. So very briefly, Clovertex, I'll give an introduction to who we are and why what we are doing today is really an important part of where we see ourselves going. Clovertex started out as a consulting organization, really focused on building drug discovery platform for biotech, life sciences, and biopharma. Our strength is in building high-performance computing applications, architecting them, building automation, and managing all the applications in the cloud. We work very closely with AWS, and our focus areas are really the discovery research and building scientific workflows in the discovery space. We do have expertise in structural biology, omics, high performance computing, big data, and analytics. We have partnerships with Intel and Veka, and we are building our partnerships with other organizations who are not only in the infrastructure space, but in the scientific domain also. So what we are going to start, and by the way, if you have any questions when we are going through this presentation, please use the question button and somebody will attend to those questions. And at the end of the presentation, we'll start answering those questions. Toshio-san, if you don't mind, very quickly, can you talk to what single particle analysis is and why it is important?
Okay, Devin. So I will just review and go through the general principle and protocol of the cryogenic electron microscopy, so-called cryo-EM. And actually, what we do is the most popular method is single particle analysis. And for this, first, we need rapidly freezing the solution of the protein. As you can see from the left top, there is a solution which contains a bunch of the same kind of protein. And this is what we need as the start point. And we apply the drop to the grid and plunge into the liquid ethane for the rapid freezing so that the water won't be crystallized, but vitrified, means like glass-like kind of the state. And inside of this, the protein will be embedded so that it will be protected from the vacuum condition, so that the protein won't be broken by the vacuum condition. So the cryo-electron microscope here that we show the red rectangle is actually a very special transmission electron microscope, where with which we can keep the specimen in very cold temperature, which is usually we use a liquid nitrogen temperature, so that the sample, the damage to the sample by electron beam will be reduced compared with room temperature. Using this equipment, we can get these image we called microglass, and recently it became possible to take movies of microglass. And each microglass contains many protein particles, which is and all the particles belong to the same kind of protein. That's why we call it single particle. And then to recover this micrograph is in 2D image, actually more precisely 2D transmission image. So we have to somehow reconstruct or restore the 3D structure of the original particle from the 2D images. For this, we use computer image processing, and this method is called 3D reconstruction procedure. Once we get the final high-resolution structure or map, then, next please, we can start building the atomic modeling based on this 3D map we obtained from cryo-EM. So my actually research focus is computer image processing part of single particle analysis. So it's based on the heavily statistic, math, image processing, and 3D reconstruction algorithm. So, for example, this is an example of the typical computational workflow used in cryo-EM single particle analysis. This is from the software called Relion, and this is the typically used protocol, simplest, but it works. So there is, as you can see, there is many steps. Like at least this is a dozen. And each algorithm does a very different image processing task. And so cryo-EM is one of the methods which is the computational part is very heavy, especially compared with X-ray crystallography and NMR. Thank you. Next, please. So we try to basically, we want to automate, of course, because because that the image processing is so heavy and complicated. Currently, that the people who want to use this method have to learn very intense or deep theory. Try to use this method and get the really high resolution structure map. But this is not practical. So what Clovertech and Dr. Kihara and me are trying to do is we want to automate this procedure. So MyPad and Clovertech are trying to do automation of the workflow from cryo-EM image aggregation to map. So there is a grid preparation, and then we manually move to the cryo-EM and put the specimen into the cryo-EM and take picture. This part is done by the human, and also the cryo-EM image aggregation part is almost completely automated by the electron microscope makers. And what we do is after the image is acquired, then we automatically transfer the movies or the data that we acquired to the so-called on the fly process computer, which is on premise, and this is relatively cheap desktop, and it's not that cheap, but powerful desktop. But we don't need to use a cluster or computer for this. Here, that we do preliminary processing, such as motion correction, CTF parameter expansion, which is very specific to cryo-EM imaging, and then recognize each particle of the protein from the micrograph, and then cut out the small region around it and make the stack of the so-called particle image stack. And then we do more robust processing and going to the 3D domain. However, this going to the 3D domain is heavy, so we do very preliminary 3D process, but more heavy process will be done by done in the virtual cluster in the cloud computing environment, such as AWS. And for this, that we have to establish that the platform using on the cloud computing environment, so that we can actually run the software such as Reliant. And so far, we have finished phase 1. So we can do, a human can do analysis as before, but manually. Currently, we are working on fully automatic processing for compound screening with help of the Clovertech. And this is ongoing effort. So up to here, it's almost automated. And in the future, this is far future, we also want to automate the protein dynamic analysis, which that we don't cover so much today. But this is our the goal or aim of the project. Next, please.
Yeah, thank you, Satoshi-san. I just wanted to point out a couple of things here. Is that the amount of data and the amount of compute needed for doing this analysis is really high. Typically, sample produces 3 to 7 terabytes of data. It needs GPU and CPU compute. And so having access to the storage and the compute, cloud is a very good environment to run this analysis on.
Right. Thank you very much, Devin.
Yeah.
Yeah. The abstract of the automation is currently human intervention. So we have many steps. But for the each step, actually, still the human invention is necessary to obtain the high resolution map. And mainly it's a selection of the good and good images, 2D images, or good 3D map. What I mean by the good means it looks like protein or not. And this is not so easy for the people who are not used to the single particle analysis. So you need a certain amount of experience looking at image and gain this feeling of, oh, it looks like protein, right? So, but the problem is it takes time. So nowadays, people are trying to, since that after the deep learning became so powerful, from the recent development of deep learning methodology, so we started applying this deep learning method to our field too. So now some of the human intervention was automated by using deep learning. So this is the example. The right side is example. For example, particle picking. There is many of the algorithm developed by the many research group, and nowadays the Topaz and Cryolo are most used ones. But these are powerful enough so that actually we can generalize model, neural network model, and at this point, almost no human intervention is involved for picking the particles. Only if the data set is not so unique. Sometimes you need human intervention, but most of the time, I say 80% to 90%, no human, you don't need to do anything to pick the particles. The other one is a 3D construction of initial reconstruction, 3D classification, and 3D refinement. So these are more like the heterogeneity analysis, meaning that actually to capture the motion of the protein inside of a data set, because the protein have to move to exert its functions. The other one is post-processing and also atomic modeling. And especially the atomic modeling, I think Dr. Kihara will cover a story about his effort of developing these algorithms using the deep learning methodology. Next, please. So, about automation, going back to the automation. So without such an elaborate deep learning section, there is primitive and fast automation done by Relion Group. And we call it on-the-fly processing, which means that while we are taking picture one by one, we can process each movies or the microglass movie that one by one. So the early point of the process, early stage of the processing can be done like this. So we do some pre-processing while we are taking picture, which reduces the time it necessary to finish the whole analysis procedure. So this is called Relion 8, and as you can see, these are very simple, but you can get a preliminary 3D map, and so that you can check the quality of the data set or sample. Next, please. So we also to expand this, that we made a customized Relion 4 scheme for compound screening workflow. So compound screening means that so for the structure-based drug design, abbreviated SBDD, you want to get structure of about hundreds compound which are bound to the target protein. And therefore, that we are repeating the similar procedure and getting the very similar structure. The only difference is this small compound, meaning the small molecule, is whether attached to the protein or not. And if it's attached, how it's kind of the posed relative to the protein. And this is very helpful to do the drug design and improve that choosing the best optimal small compound or the small molecules for a drug as a drug. So based on this, so the reason why that we put this specialized purpose is because it's easy to automate. So for example, if it's compound screening, usually you have already up structure map, means the structure of the target protein without any compound added. And so this data analysis that all the pharmaceutical company does first, and then that since you have a map, you can also make the reference mask or 3D mask. So we can assume these mask reference map and the reference mask are available already. Also, we assume that on the pre-processing are done. And so this is mainly for the processing on the cloud environment, because these are heavy and aiming for the high resolution so that you can see the compound, small compound. So for this, we need parameter setting per protein that user have to change. And also user have to optimize parallel computing setting per computing environment, such as cloud or on-premise the GPU box. And also it is important to be able to generate the processing time and the cost level automatically. So these are something that we are working on now at KEK. And importantly, the parallel computing setting per computing environment can be very complicated for the AWS. So we are doing lots of benchmark, many benchmark testing, so that we are choosing the optimal machine type and also the parallel computing settings for now. Next, please. So after we did the, So we learned the CS schemes, which the previous, I show you the previous slide. We learned that with some data set and started to find the problem. So the 1 and 2, 1 is centering your particle and 2 beta automatic selection is necessary. So as you can see from the picture, the particles are not really centered. So for example, the bottom part is actually that including the 2 particles instead of 1 particles of the same protein. So if it's centered well, it looks like the top part of the protein. So it's just 1 protein. But since the centering is not good, that the particle picker is picking that the 2 particles together is 1 particle. And this is a problem. So we had to do something about it. Beta automatic selection is that selecting based on the 2D and 3D correction. As you can see that the red part is what automatically selected particle image, but it contains some of the non-desirable particle image, such as one contains the 2 particles together in 1 image, or the one which looks like the very bright spot. These are not really desirable to include in the further processing. So 3 and 4 are also important. So we find out more optimization is necessary, especially that we need to do the maybe improve the workflow design, also job parameter settings, or meaning the algorithm parameter setting and blah, blah, blah. Also, the cost reduction is important. So we, especially that we need that really think about target resolution and cost and all other. So we need the optimized basically balance among the resolution and speed and cost. Importantly, the result shows the CS scheme achieve better resolution than non-expert, which is actually the 5 years ago me, because this data set I did analysis that do the single particle analysis manually. And it was 5 years ago, and we could publish it. Could you go back? And then, so basically this means that the well-designed workflow and optimized parameter setting by Cryo-EM SPA expert, which is current me, it does really better than manual procedure often. So this indication was very encouraging for us. So we will continue working on this and do the more optimization and the cost reduction. Next, please.
Yeah, thank you, Sulosha-san. This is really going to show our audience that some of the decisions we made in the automation of SPA particle analysis as well as CS analysis really improves the performance, and potentially there is a potential for optimization as well as cost reduction. Thank you so much. Again, if there are any questions on this section, please use the Q&A button, and we'll certainly get to those questions. Thank you so much. Now we are going to shift our gears and start talking about atomic model building. So the whole goal of doing the cryo-EM analysis is really get to an atomic model for the protein. We do the cryo-EM experiment. There is a huge amount of data which is really coming in. If you look at the data submissions in any of those public repositories, there's a huge amount of data coming in. But really, we want to go to the atomic models associated with that structure. Just an example, here's a graph we have picked up from EMD, where what I'm showing here is on the x-axis the year, and on the y-axis the number of entries released per year. But if you notice it, a lot of the effort is going in getting to the resolutions around 3 to 4 angstroms. That's where a lot of the analysis is focused on. But there is a fair number of data points which have so-called worse resolution, in the sense that the number higher the number, the worse is the resolution. And Dr. Kihara and his lab is actually working on the models which will allow analysis of the lower resolution data which is coming in. And we'll actually talk about that very quickly. Ultimately, the goal of atomic model building is really to build a model, hopefully with resolution better than 3.5 angstrom, which is where cutoff today is. But it typically requires a huge amount of time as well as high level of expertise, including a lot of compute to do it manually. And there's a lot of activity happening in using machine learning, including deep learning in model building. What model building needs is the density map, which we received from the cryo-EM. But you want to be able to bring additional information which you have available for those proteins in question, and bring that knowledge to get an atomic model which is closer to reality. Here's a list of tools, and this is a very small list of tools. One tool I'm highlighting, and these are all based on neural network tools. The EMAP2-seq has been developed by Dr. Kihara Lab. And what today he's going to talk about is not only the work which he's doing, but what he sees in this field his lab is doing, how they are progressing, and what kind of tools and thinking process they have in building atomic models. Dr. Kihara.
Okay, thank you, Devin. Now I'll show you, introduce a couple of tools we have been developing by using the deep learning. So the first tool, which will be very useful for structural modeling, is this deep learning-based quality estimation scores, DAQ. What DAQ does is it is used for in the process of protein structural model building. DAQ checks the local density pattern of a protein model you are developing, the pipeline is developing in the process, and then checks if the local density pattern learned from the known cryo-EM maps from EMDB agrees well with the residue position, each residue of the model or not. So you can actually, so this is very useful because it's very typical that the mistakes can happen when the sequence in the sequence mapping stage, for example. You may have a nice alpha helix, but the sequence mapping could be wrong. Then DAQ can detect those kind of errors. On the left shown is the database called the DAQ score database. You can probably check later. And in this database, we analyzed over 100,000 of protein chains currently stored in PDB database. And you can see that some of the models may have errors in it. And then you can take a look at it to help us to see how DAQ works. Then the next slide is about, now I'm going to the structure building process. The first one called cryo-read. This is for RNA and then DNA structure modeling. This has been recently published. So what it does is it's an automated process for building DNA or RNA structures in the cryo-EM. So I on the left, I quickly walk through how the algorithm works. In the first step, it uses deep learning to detect the position of sugar position and phosphate position and base position in the map. And it also predicts the base types. When the map resolution is up to around 5 angstrom, then we can detect those features, those are the structural element in the cryo-EM map sufficiently accurately. And then in the next step, those sugar positions are connected to build up the chain, main chain of the nucleic acid. And then after the structure is built, then DNA or RNA sequence is mapped to the structure by matching your sequence with the detected base assignment, and then do the refinement. And a very important thing for this pipeline is that this is fully automated. Actually, there's a parameters in it, but this pipeline, this cryo-read, will use basically all the combination of parameters inside, and then at the end choose the best model by checking the score. So on the right, I'm showing just 2 examples. A is for DNA structure. In the middle shown is the structure in PDB. On the right are what cryo-read generated by automated fashion. There's no human intervention, no parameter tuning, but you get this structure just out of this software. B is more complicated structure. Green is on the left is the ground truth. This green is proteins, and it is complex with RNAs, and many chains of RNA can be fairly well modeled. And that will be included in this Clovertex pipeline. Okay, so the next one is now about proteins. So we use the same logic here, pipeline. This is now for proteins. You have now I walk through on the left diagram from the top. You have a cryo-EM map, and then deep learning will detect amino acid positions and C alpha positions and amino acid type, and then detected structural elements is connected, assembled into fragments, and then those fragments are finally assembled into the full protein structure. Important thing we implemented is actually AlphaFold. As you know, that AlphaFold has been very successful in structure modeling, and that is also very useful for cryo-EM structure modeling. So we do the AlphaFold, but as I show you in the next slides, AlphaFold structure is not always perfect. Actually, there are small shifts, and then domain orientation can be very wrong in AlphaFold models. So what we do is that we use AlphaFold model, but cut the model into pieces, and then we use the fragment from AlphaFold. Fragment is integrated to the library from the main chain trace, and then that is combined and then used to build up the entire protein structure. We can also do the protein complex structure modeling. In the next slides, I show you a couple of examples. The diagram on the left is comparing performance against AlphaFold 2. AF2 is AlphaFold 2. Y-axis is deep mainmast. And then what is shown is that, okay, in comparison, deep mainmast is having a higher score for 157 cases, and AF2 was better for 21 cases in my benchmark data set. So on the right the examples. This is comparing with our score deep mainmast against AlphaFold 2. So okay, so the bottom part is AlphaFold 2. What you see is that in AlphaFold 2, although the individual protein domains are well modeled, actually typical on the left, the alpha helices in AlphaFold 2 model on the bottom are slightly tilted, but which does not fit to the density. But our method's deep mainmast is tracing the density, and that's why the alpha helix are very nicely fitting modeled in the map. In the middle example, it's a two-domain structures. AlphaFold 2 built the individual domain well, but relative orientation is not good. On the right, there's a long tail of the beta sheet, and AlphaFold 2 does not make this to the beta sheet at all. That long tail is merged into the main domain of the structure, but the deep mainmast can build this nice long beta strand because it is really tracing, looking at the density and tracing it. Then this slide is about multi-chain modeling. The deep mainmast is also has a specific algorithm to connect the multi-chains correctly. It's not an easy problem, especially for homo multimer cases, because the sequences are the same between different chains. But deep mainmast has a specific algorithm to connect the chains in a very natural way. Okay, and I think that's all I have.
Yeah, thank you, Kiara-san. That's extremely valuable information for not only showing where your lab is going, but how it is making an impact on the research. Not to mention that there are other tools available. I'll walk you through just one quick tool which is being developed by the Relion group called ModelAngelo for atomic modeling. They basically use the cryo-EM map, add the sequence encoding, and essentially they use GNN to build the C-alpha atomic model. We have done some experimentation on bringing ModelAngelo. What we find out is that if the resolution of the cryo-EM map is good, in the sense of it is 3.54 angstroms, it maps well. So the RMSD for, say, 6KNF is roughly 0.27, which is fantastic. But there are some proteins where the resolution is bad. The ModelAngelo actually predicts the prediction is not really all that well fitted. The RMSD for this particular compound was about 35 angstroms. But this is great information for the science in terms of how we can do automation. And Clovertex is working very closely with KEK and Purdue University to bring this into using cloud as a solution. again, if there are any questions about this section, please use the question and answer button to put your questions down, and we'll get to them shortly. The reason why we are using cloud as CloverText and using this application, I alluded to some of it. The amount of data and the amount of compute needed is extremely high for this kind of analysis. But we don't want the scientists to really worry about the infrastructure, which is behind the scene needed, the amount of storage and the amount of compute. So we want to build a platform which allows the scientists to get to their analysis and results very fast. We want to optimize it for cost. There is a perception, ill-conceived probably, that cloud is expensive, but we have built tools within our platform which allow it to manage and optimize the cloud for storage and compute. We want to bring all the tools, applications, and workflows to the cloud. So this automation workflow pipeline is very amenable to the cloud. We want to bring it in a collaboration environment so that people can share the results and data if they want to. But over and above, we want to have it secure so that all the data and privacy and access controls are already built in. And we want to use the scale of the cloud for building all the analysis effectively and use it as we want to use it. So this is just a very high level end-to-end workflow orchestration we are putting together for automation, both for the cryo-EM analysis as well as atomic model building. And if people have questions, they can really reach out to us and we can walk you through these workflows. Very quickly, why are we doing this? We basically are HPC experts, high performance computing. We do build a lot of applications in the high performance compute. And we do have a lot of the aspects of managing the cloud, managing the scientific computing, really under our belt to make sure that it is optimized and it is effectively used. We do have scientific domain expertise. We have people who were scientists in the organization. They have converted into technologists. They have used applications in their scientific journey. They are knowledgeable across the entire value chain of life sciences, informatics, drug discovery, etc. And of course, we have a large amount of experts from the technology side who are experienced in building, maintaining these environments and workflows in the cloud. We have the ability to scale using the most options in the cloud. We have a global presence, and we have the ability to deliver at scale. And over and above this, we not only build the applications in the cloud, but we have the ability to support operationally as well as from the functional perspective in the cloud.
Okay, thank you all for a very informative presentation. And we do have some questions already, which we'll be getting to in a moment. If you've not asked a question yet, it's not too late. Just enter them in the Q&A button at the bottom of the webinar window and type in your questions. Also, if you'd like to contact Devin directly after this webinar, his contact information is on the screen. Also, if you'd like to stay informed about the latest developments around these topics, we encourage you to follow CloverText on LinkedIn, and there is the address there. Okay, so now on to the questions. First, we have a question from Ed asking, related to slide 11. What programs or custom scripts are you using to automatically move data from camera to on-premises live processing and from there to the cloud?
So we developed our own script, and basically that we have the detector server and also the transfer server, so the detector server won't be cracked. So we will just dump all the movie as quick as possible from the detector server. Then from the transfer server, we send to the raster, the parallel processing file server. And the script is mainly using the rsync at this point. And so that it will basically check the status of the file saved inside of a server. And then whenever it finds a new file, that it will transfer automatically. So it's a very simple script that we developed for that purpose. Thank you.
Okay, thank you. All right, a question from Simone, who says, related to the custom Reliant 4.0 scheme, can the map of the APO structure be generated by another software, or should it be done also in Reliant to keep the grayscale level?
So is it again me? So sorry, could you repeat the question?
Absolutely. So related to the custom Reliant 4.0 scheme,
Yeah.
can the map of the APO structure be generated by another software, or should it be done also in Reliant to keep the grayscale level?
So that there is a setting that's whether the in the Reliant, whether the map is absolute grayscale, which that the Reliant expects, means that the normalization procedure is same as Reliant or not. And in this case, that you have to tweak that part if you want to use the map from different software packages. And also that you have to also create a script to do it automatically if you want to automate it. So yes, in the future we are thinking about incorporating that some of the tool, external tool, other the software other than Reliant, such as CrySpark, if possible, as soon as possible, actually. So that's what we are thinking for the automation, including the many software packages.
Great, thank you. Here's a question from Sonia, who asks, I might have missed it. It's not entirely clear to me to what extent the computing settings are currently selected by an experienced person or automated. I assume the goal would be to let the computing cluster decide itself, based on 2D pre-processed data, how to allocate its resources for each dataset or sets of datasets from one compound screen.
That's interesting question. Yes, it's desirable, but is it possible? Yes, it is possible. Yeah, I haven't given a thought about it too so much, because we wanted to make this system as quick as possible. So we couldn't have a chance to give a thought to automate the parallel computing setting yet. But yeah, I think it's possible once we gain the enough experience and also the time to implement those kind of things. Does it answer the question?
Great. Great. Thank you. Another question, is centering of particles automatically optimized? How is it done?
We haven't. And that's a problem. So at this point, the best way that the centering the particle for me is using the either the 3D classification or the 3D refinement using basically 3D procedure, because I believe the best center of the 2D image is the same as center of the 3D. So that's what we do, but it's very, very heavy process. So that we are trying to develop the more cheaper way of the centering the particle, especially if we can keep it the process within 2D domain, that'd be the best.
Great. And we have a couple of questions from Vadim, who says, thank you for sharing your experience. Regarding compound screening, what is the typical number of micrographs that you need to get good resolution of the ligand? Given the cost of running cryo-EM, even with all optimization, I think checking more than 10 compounds is prohibitively expensive.
Yeah, I totally agree with you. And this is the undergoing effort for us too. So at this point, we start from the 200,000 particle image and check whether we can gain we can reach to the resolution under the 3 angstrom, because under 3 angstrom you should we can see the pores and the relatively clear shape of the side chain. so that we can more identify the ports and also shape of the compound very well. 3.5 is a little bit low. I mean, you can see the density often, but the pose is a little bit difficult to determine with high confidence. So we are aiming 3 angstrom, and we start from the 200,000 particle images. But of course, it depends on many parameters, such as the symmetry, for example, and also that the thickness of ice, which can be valid even though you are taking that the same, exactly same the sample with almost same condition. Still, you know, if you just breathe it onto it, then you get like, you know, lots of that the crystal the ice contamination on top of the surface of the thin membrane of the vitrified ice. And so these kind of components, we can't avoid it. So we are trying to get the statistic by running the many data set at this point. And so far that 2000 seems to be okay. Starting from 200,000 seems to be okay, but 100,000 seems to be a little bit too low. And some of them, we need a little bit more to reach to the under 3 angstrom. So unfortunately, I can't really answer concrete, give you the concrete answer, but that this basically, please remember that this is ongoing effort. So I think that I hope that we can answer this question more clear way near future.
Great. Thank you. Another from Vadim. How does the DAQ score compare to the MAP-Q score, which was recently adopted by EMDB as the quality measure?
Okay, I will answer this question. So the DAQ score uses deep learning to see the amino acid specific local density. In contrast, Q score is checking the overlap between the atom and then the volume. So the density. So what can happen is that, you know, if an atom is placed in the density, but if the identity of the atom, like amino acid, is not incorrect, then DAQ can detect it, but Q score does not.
Okay, and one last from Vadim. Can cryo-read handle protein nucleic acid complexes?
Right. So cryo-read is only for nucleic acid structures. So the pipeline we are developing is that first we use EMAP2-seq, which was in the list of the software Devin has shown, to detect a DNA region or a nucleic acid region and protein region, and then apply cryo-read and then deep MAME-MUST for individual protein region and the DNA RNA regions. Actually, the DNA RNA region can be detected with very high accuracy, almost more than 90% accuracy at the resolution level up to 10 angstrom. So and then we are currently making this pipeline for this.
Great, thank you. Here's a question from Shiv. How to proceed with model building of a protein cryo-EM map of 6A resolution? Right.
So this is a good question. So deep MAME-MUST cannot see the deep learning cannot see the amino acid position precisely at 6 angstrom resolution. However, so what we have is a pipeline of doing AlphaFold. So what we do is that first we do the AlphaFold to the protein sequence, and then detect the individual domain of the structure, and then do the fitting to the cryo-EM map. We have a specific software called Vesper, which does the good superimposition.
Great, thank you. A question from Nemul, hopefully I'm pronouncing that correctly. For cryo-read, what resolution is required to build the nucleic acid? Does it require to provide DNA or RNA sequences as input?
Yeah, good question. With the same reason, cryo-read needs resolution up to 5 angstrom currently. But you don't need, yeah, if you have a sequence, yeah, we can map the sequence. But if you don't have the sequence, we just make the main chain structure of DNA RNA. So sequence is not necessary, but if you have it, we can map it.
Great. Another question from Sonia. Can I ask up to what AA chain length deep main mast can build a structure reliably? And what is the minimum map resolution needed to build a reliable model?
Okay, so the size of the protein actually depends on how much memory we put on the cloud. We can easily do more than 1,000 amino acid chain, and we can do more depending on how much memory size we put on the AWS pipeline. And then minimum map resolution we would need is up to around 5 angstrom. Because above 5 angstrom, beyond 5 angstrom, amino acid type cannot be detected very precisely.
Great. Question from Pierre Damien. Is deep main mast working with nucleic acid protein complexes such as ribosomes or only pure protein complexes?
Right. So I think this was the same question. Deep main mass is only for proteins. So first, if you have a complex structure between a protein and DNA, RNA, first we detect DNA region, RNA region, and the protein region, and then apply deep main mass and cryo-read separately to each regions.
Got it. So Simone asks, is deep main mass doing only modeling or only refinement of the structure?
Right. It's for modeling. It has it's for a de novo modeling from the cryo-EM map. If you already have the good structure, it's good for you to use a conventional tool like MDFF or Rosetta to do the refinement.
Got it. Here's another question from Debu. What do we need to do to get a demo of the platform?
Okay, so if it is only for a protein modeling part, my part, we have our lab has a website to do the modeling, and you can come over and then test it. We have that also source code made available, so you can also download and test it. Well, we can help you to do it.
Thank you. All right, that's all the time we have for questions at the moment. Although we have all the questions noted, we will be following up with answers on all of them. And we'd like to thank everyone for attending the webinar. Thank you and have a great day.